All right, chapter 15 is the immune system. You guys in micro, we're going to hear this again. Pretty much same stuff. But that's okay. It helps it stick in your head. So we've got a couple of terms to define here. One, and we've defined this already. We've already defined this, but I want to define it again because immune cells secrete tons of these. So cytokines. We're going to talk about a bunch of cytokines here. Cytokines are chemicals released from cells that cause an action in another cell. And the immune system releases bunches of these. Bunches. And they usually call them by names like IL-1 and IL-12. IL stands for interleukin, which means in between white. So white blood cells talk to white blood cells. White blood cells, those are the interleukins. And then other cytokines are going to cause blood vessels to be leaky. And we've used this term, so we're going to use it more here. An antibody is going to be abbreviated AB. And it's a protein produced by B cells in response to an antigen. Antibody, and you've heard about antibodies. I know you've heard of antibodies. Antibodies are produced by B cells in response to an antigen. When we talk about the antibody there are specific types of antibodies. And they're in classes, and there's 5 classes. So when we talk about them generally, they're called antibodies. When we talk about them specifically, then we're going to call them immunoglobulins. Immunoglobulins. And we'll describe these in a second. An antigen is going to be abbreviated AG, and that's anything that initiates an immune response. A virus, a bacterium, anything foreign, fungus, a protozoan, anything that we learned about in micro is potentially an antigen. And a pathogen is anything that's capable of causing disease. So, antigens don't always necessarily cause disease, but they can, like those surface proteins that are found on red blood cells, those A's and B's, are often called antigens, because if you're A blood type and somebody gives you B blood, your immune system sees that as foreign and it will initiate an immune response, right? Those proteins on the red blood cells aren't considered pathogenic, but they initiate an immune response. So, pathogens are disease-causing things, like bacteria and viruses and such. Bacteria are single-celled organisms. You're going to learn all about them in micro if you haven't yet. Single-celled organisms, they divide on their own. We have lots and lots of bacteria that live in us and on us, and they are not pathogenic unless they get in the wrong place. If they get in the wrong place, then they become antigenic, and they can initiate an immune response. Viruses are comprised of just RNA or DNA and a protein coat. RNA or DNA and a protein coat. Bacteria can divide on their own. You can put a bacterium on a petri dish and it can divide. Viruses cannot. Viruses are just nucleic acid with a protein coat. So the way they divide is they have to get inside your cell, and then your cell gets confused and makes copies of the viral pieces and parts, and then they get put together, and then often what happens is the virus blows the cell up. So it uses the cell to make copies of itself and makes many, many copies, so that when it's released, those viruses go to infect other cells, which make many, many, many copies, which then explode a whole bunch of cells, and then we're in trouble. Prions are infectious proteins. These are kind of weird. We all have normal prions in our cell and on our cell. Prions are proteins. And what do we know about proteins? Shape is vital to function on proteins. So these normal prions have specific shapes, right? And specific shapes mean they do their job. So if you're exposed to abnormal prions, either because we don't know why, or you ate bad beef, or some prion diseases are genetic, then you get in your body abnormally shaped prions. They sit next to your normal ones and cause them to change shape. And when they change shape, they lose their function, and then the cell dies. And so do you. Prion disease is 100% deadly. It will kill you. All right, so those are just some of the pathogens that you're going to learn about in micro. Antibodies, we're going to go back to antibodies. So an antibody is a Y-shaped protein. They are produced by one of our lymphocytes called a B cell. Produced by one of our lymphocytes called a B cell. We'll talk about them again. We have the capability to make antibodies to any potential antigen. So here's a pathogen. Here's some kind of bacteria. And on the surface, it's got all these different structures. Because they're going to initiate an immune response, they're considered antigens. They look foreign to my immune system, and I can make antibodies that look at this one and can make that shape. I can make antibodies that recognize this antigen, and I can make antibodies that recognize this one, and I can make antibodies that recognize that one. We have the capability to make antibodies to anything we come across. The antibody itself has a, I don't know, it's the legs. The legs of the antibody are constant. In other words, all antibodies have the same structure. The arms are the same as well. It's the hand portion that changes. So all antibodies are the same from here on down except for the hand portion. It's the hand portion that binds to the antigen. So you can make antibodies like this, or this, or this, or this, or this, or this. What haven't I done? This, this, okay, sorry. This, this, this. Get it? Mm-hmm. So we should be able to bind to anything that we come across. Which we can. If our lungs or immune system is healthy, we should be able to at least mount in a response to anything that we come across. When we talk about antibodies collectively, I've got high antibody levels. Well, that means we're looking at all these different classes of antibodies. Potential. When we talk about the antibodies individually, we talk about them as immunoglobulins. Sorry. And we're going to abbreviate them IG. IG for immunoglobulin. These are all antibodies. They're all a type of antibody. IgG antibodies make up the most abundant antibody in the body. Antibody in the body. Most antibodies are the IgG type. Like 80% of your antibodies are IgGs. We find IgG antibodies in, they're important in what's called the secondary response. Secondary response. In other words, you get sick, you fight off an infection, you have antibodies floating around, and those that are floating around looking for that pathogen again are of the IgG type. The IgG. This is what gets increased after vaccinations, and it's part of our immune memory. IgAs are found in lots of different secretions, like in your tears and in your saliva, and in mother's breast milk. So you've probably heard if you breastfeed, then you give your baby immunity. Well, that immunity comes from these IgAs. So it helps with and sort of GI type things because it's more in the gut as opposed to anywhere else. IgE, these don't last forever though. As soon as you're done breastfeeding, these die out in about 2 weeks. But by that time, baby's been exposed to other their own stuff and they're building their own immune response. So it's kind of a little extra. IgEs are part of allergic reactions. IgEs are part of allergic reactions. So an IgE antibody stands on the surface of two types of cells. Well, basophils is one of them. And another cell called a mast cell. Basophils, remember, have big granules in them. So do mast cells. So if this IgE is allergic, if you're allergic to pollen and you breathe in pollen, then that pollen binds to the receptors on the IgE that's standing on this B cell, and that causes this basophil or mast cell to release their granules, and those granules have histamine in them. And histamine gives you the allergic response. It makes your blood vessels leaky and swelling and itching and redness and weeping, stuff like that. IgMs are part of the primary response. In other words, when you first get sick from something, you make IgM type antibodies. IgM type antibodies. The ones that are going to hang out for later exposure are the IgGs. IgMs are interesting in that they're very large. So they all kind of come together like a big snowflake. Those are IgMs. IgMs are also in the antibodies for blood groups. They're the antibodies that you find in blood groups. So if you are A blood, you've got what kind of antibodies? B's, and they're this kind of snowflake shape. That's good, because if mom is, now remember, if you're given B blood, then your antibodies will bind to the B blood and target it for destruction, right? But if you are pregnant and you are A blood and baby's B blood, how come mom's antibodies to B don't damage baby's blood? Because these are too big to cross the placenta. So baby is protected. This big snowflake structure can't get across the placental lining. So baby's okay. That's IgMs. IgDs, they're not exactly sure of the functions of the IgDs. They think that they stand on the surface of some of your B cells and just act as a receptor. They're not too sure about the IgDs. All right, got to review our little white blood cells again. We talked about this before. We looked at the structures and you kind of mentioned this. Monocytes have the big horseshoe nucleus, solid horseshoe. They circulate in the bloodstream as a monocyte. So here we are in the capillary. Here's the cells in the capillary. When you have infection, when you have damaged cells out in the tissue spaces, then the cells that are damaged, or these or the cells that are already there, well, let's do it this way. If the cells are damaged, so we've got damaged cells here, they've been blown up or whatever, they release chemicals called cytokines, certain cytokines, and that makes the monocyte attracted to them, and they head to the edge of the capillary, and they squeeze through the capillary, and then they turn into that macrophage, M with an O through it is our macrophage sign, and macrophages are phagocytic. They eat stuff. They eat junk and debris, they eat dead cells, they eat bacteria. but they are not phagocytic as a monocyte. They're only phagocytic as a macrophage. They look different, their job is different. And then once they've done their job, they'll undergo apoptosis and they'll die. So they don't live there forever. Lymphocytes reside mostly in our lymphoid tissue, like our lymph nodes, our tonsils, our spleen. And the two types of cells that we're going to work with in this chapter are T cells and B cells. T cells and B cells. Now, B cells are the one that make antibodies. B cell makes antibodies. B for B. Then we've got our granulocytes. Worked with these in lab, right? Our three granulocytes include our neutrophils, our eosinophils, and our basophils. We know what neutrophils look like because they got that big pinchy nucleus. Eosinophils also have a pinchy nucleus, but they've got those red granules. And then the basophils have our big blue granule. So eosinophils are neutrophilic, are phagocytic. They're the first to arrive to an infection. First to arrive, and they love to eat bacteria. They love to eat bacteria. It's their favorite meal. So they get to an infection site before monocytes or macrophages. Eosinophils are for allergies and worm infections, and basophils are allergic reactions. So, a little bit of review of our white blood cells. So when a white blood cell I like this picture. I like this. Looks like I found it online. Here's a phagocytic cell reaching out and engulfing, so it reaches its cell membrane out and surrounds bacteria, pulls it in. Now it's in a little membrane-bound sac. Fuses with a bunch of lysosomes. The lysosomes have those enzymes that chop up the contents, and then they are exocytosed out the other end. So phagocytic cells include macrophages and neutrophils for the most part. Eosinophils can be phagocytic, but not very often. The last type of white blood cell we're going to talk about is called a dendritic cell. A dendritic cell is an antigen-presenting cell. Antigen-presenting cell. And what it's going to do, we talked about this, didn't we? Yes or no? Was that, no? Maybe it was anatomy we did this. I think it was anatomy. I'm getting, now I'm doing all this stuff in all the classes again. So dendritic cells are phagocytic cells. Whoops. They're phagocytic, but what they do, instead of taking this in and taking it to the lysosome and chopping it out and spitting it out, they take it in and they cut it into little pieces. They cut the pathogen into little pieces, and then they stick the piece out on the cell surface, and then they run around and present it to different white blood cells, specifically your T cells and your B cells. So they don't necessarily get rid of that, but then they leave their home. So we've got dendritic cells that live in our tissues, like in our skin and our lungs and our other places. And so when they find something, they eat it, and then they chop it up, they stick it out on the cell surface, and then they leave. They enter the lymph, because that's where your lymphocytes live, your T cells and your B cells, and then they show it around and see if somebody would like to bind to it and start an immune reaction. So this is where we're going to fight one thing at a time. Oh, we started this in micro, didn't we? That's where we did it. I'm like, I am talking the same story here. Why am I deja vuing? I wonder if my things are going to work.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the
This is
antigen originates
phagocytosis
outside
and
the cell.
dendritic
Phagocytes,
presentation.
such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class 2 MHCs, and the two structures combine. The MHC class 2 antigen complex is transported to the plasma membrane. The displayed MHC class 2 antigen complex can stimulate other immune system cells to respond to the antigen.
Let's see, what do we talk about MHC? Not till here. There's a slide on the mic in micro that I like, so I'm going to replicate it here. The immune system is divided into two sides. The non-specific side, which is called innate immunity. We've got a specific, which is known as acquired or adaptive. I like to use specific. I like non-specific and specific because it helps me remember what we're dealing with. Within these, we've got some more subdivisions. These are known as our lines of defense. So we've got a first line, we've got a second line, and a third line. First and second line of defense. So non-specific immunity is going to be our immune response to it's going to be a non-specific response to an infection. In other words, we're going to fight off multiple potential pathogens that might attack us. But first of all, we got to talk about our first line of defense. So the first line of defense are the physical barriers. In other words, what keeps these pathogens out of us? And it includes the skin. Skin is a perfect barrier. It's many layers of dead cells. It's dry. It's a little acidic. As long as it's intact, it's perfect. We have mucous membranes. Mucous membranes are any tube that opens to the outside, makes lots of mucus, thick mucus, so that if you are infected with, say, a virus into your respiratory tract, in order for that virus to make copies, it's got to make its way through the mucus and bind to your cells. So that mucus acts as a barrier. In some of our mucous membranes, they are also ciliated. So like in your respiratory tract, if you remember from anatomy, we've got cilia that beat upward, called the mucus escalator, and that moves the mucus now. So now if you've got pathogens, viruses, or whatever, bacteria in there, and it's moving, it makes it harder for them to get to your cells to bind. So bacteria or viruses have to bind to your cells before they can either divide or get inside the cell. We've got acid, we've got stomach acid. That's a great barrier. Nothing you eat is sterile. So we eat stuff all the time that is potentially hazardous. But because of that pH of 1, we denature proteins. Pathogens die. So these are some of the things that are keeping these pathogens out of us. And those are very important. As long as they're intact and they're doing what they're supposed to do, we are okay. Now, if your physical barriers get broken, then we're going to move on to the second line of defense. Now we're going to move into the inflammatory response. Inflammation. This is where some pathogen has gotten in you, and we're going to mount a local immune response. An immune response right in the little location where we've got the infection to try to get rid of whatever's potentially there. So when let's say your tissue, your skin is cut, or your lungs are dried and cracked, or your tissues or whatever, the damaged cells are going to release cytokines, and those cytokines include histamine and interleukins, which we'll talk to other damaged cells release cytokines. Macrophages release these two. So the damaged cells release the cytokines. Those cytokines call for your macrophages and your neutrophils to show up. Macrophages and neutrophils. Makes blood vessels leaky. When macrophages get there, they start eating things. And as they eat bacteria or viruses, they release more cytokines that calls for their friends to come, increasing the temperature of the area, maybe even giving you a fever, increasing capillary permeability even more, so that those macrophages and neutrophils can get out of the blood vessel easier. And then we also results in our swelling. So here we've got skin that's been poked by a very, very dirty pin or a knife or whatever. So our physical barriers have been broken. Pathogens have entered. Damaged cells release chemical signals. Those chemical signals attract macrophages, monocytes, right? They attract the monocytes and they attract the neutrophils. They squeeze out of the capillary. They start eating the bacteria that they find. They release more chemicals that call for more friends to come and join the party. Now we've got a whole bunch of phagocytes running around eating, in this case, all these bacteria that came in. Of course, we've got clotting that goes on and all that stuff too to stop the blood flow. So the inflammatory response involves macrophages and neutrophils, which are phagocytes. Macrophages and neutrophils, these are their phagocytes. They don't care what bacteria that is. They don't care. They don't care if this is Staph epi or Staph aureus or E. coli or whatever. They eat whatever they find non-specifically. It's the non-specific response. Whatever they see, they go out and chew it up, hoping to keep this little infection in a local place so that we don't have to bring in our third line of defense, which is the big dogs. Those are the big dogs. So, the key here for the second line of defense is that it is mediated or controlled by or ruled by phagocytes. Phagocytes rule. Phagocytes run. Phagocytes are in charge of the second line of defense. Neutrophils and macrophages. And they don't care what they find. They eat non-specific specifically. They eat whatever they find. It's the third line of defense where we're gonna start targeting one infection at a time, one thing. Second aspect to the second line of defense is something called complement. So we've got the inflammatory response as number one, and then we've got something called complement. And complement system is a series of 9 proteins that float around in the bloodstream, and they're activated either directly by bacteria or by bacteria that has been surrounded by antibodies. Whoops, I did that wrong. Bacteria that already have antibodies stuck to them. Okay? So, or they can bind to the bacterium directly. So, we've got these complement proteins, there's 9 of them. They come in and they bind to, let's say we've got the first one that comes in. If it binds to 2 antibodies, in other words, if it cross-links 2 antibodies, that activates that complement protein, and he calls on all of his other 9 friends, and what they do is they come in and they insert themselves in a nice little circle in the bacteria, and they form a hole. Not only one hole, but many, many holes. So they form pores in bacterial membranes and walls, and then the bacterial insides fall out, and the bacterium dies. So the complement system targets bacteria only. Doesn't really care what the bacteria is. Could be staph epi, could be E. coli, could be whatever. As long as the complement system gets activated, either by binding directly or binding to a bacterium that's already got antibodies connected to it, then all of his neighbors come in and they insert themselves into the wall and the membrane, insides fall out, and the bacterium dies. For the specific response, now we're going to talk about the specific response. Specific response has 2 sides to it. We've got a B cell side. I'll do it this way. Antibody mediated and cell mediated. Who makes antibodies? B cells. So the antibody mediated side is run by B cells. Antibody mediated side is otherwise known, kind of old-fashioned term called the humoral response. I don't like to use that because it doesn't really tell me anything, but if I say antibody mediated side, I know I'm looking at B cells. B cells make antibodies. So, we already know that B cells make antibodies. They make all of these different kinds. Some of these antibodies are soluble, meaning they float around in the bloodstream. Some of them bind to specific cells. But what antibodies do, people always want to think that antibodies are like these little missiles or grenades that blow things up, and they're not. They are not that at all. What antibodies do is they bind to antigens, and they surround them. So when you've got an antigen that is surrounded by antibodies, like so, that's called opsonized or opsonization. Opsonization. If you've got a bacterium or a virus, whatever, that is surrounded by antibodies, it makes your macrophages, your phagocytic cells find it and eat it faster. So it doesn't kill the thing, it targets it for destruction. In other words, it helps your macrophages and your neutrophils find it faster. Like, here's my favorite analogy. Oh, let's see, cupcakes, right? Cupcakes are yummy. Cupcakes are yummy. I would eat a cupcake. But if it's got frosting on it, it's even better, right? Then I'm gonna go to it. I would pick a frosted brownie or a frosted cupcake over an unfrosted cupcake any day. So think of antibodies surrounding that antigen like antibody frosting. Macrophages like it frosted. They think it's yummier, they go and they eat it faster. Does not mean they wouldn't eat it without the frosting, but it's faster with it. They like it faster, they like it better that way. Now here's the other thing about B cells. So B cells make antibodies. Antibodies' job is to surround antigens or opsonize them, put the frosting on them, so it marks them for phagocytic activity. B cells need to turn into a cell called a plasma cell. Now don't think of this like plasma of the blood, right? It's not the liquid form, it's just the name of the cell. It's a plasma cell. And technically, it's the plasma cell that makes the antibodies. But the plasma cell is derived from the B cell, so ultimately, they all go hand in hand. So B cells themselves cannot make antibodies until they turn into a plasma cell. So they've got to differentiate. It's kind of like the monocyte turns into the macrophage, right? Then he's phagocytic. The B cell turns into the plasma cell, and then the plasma cell makes the antibodies. Antibodies secrete millions of antibodies. Millions. Like 2,000 per second, just spewing out antibodies. The other thing we're going to get on this side is the B cell is going to become a plasma cell, and it's also going to become what's called a memory cell, a memory B. So memory cells will remember that we've seen this before, and then the response will be a whole lot faster. Start the T cell side. T cell side, this is cellular mediated immunity, and we've got some T cells we need to talk about. One is an NK cell, stands for natural killer. Now, natural killer cell technically lives over here because he's scouting, but he's a type of a T cell. So I always talk about him here. NK cells, we've also got a cell called a CTL, or a cytotoxic T lymphocyte. And then we've got the third, which is the T helper cell. T helper cell, or helper T. So let's do this first. I gotta define the major histocompatibility complex. The major, and they already mentioned this in that video, major histocompatibility complex, or MHC, is a complex group of proteins that lives on all nucleated cells, so not red blood cells, they don't have a nucleus anymore, but all nucleated cells, and it defines you as you. It defines you as you. This is a complex group of proteins, and it's found on these genes to make this protein are found on chromosome 6, found on chromosome 6, and there are many, many different forms of it. So, let's see if we can look at this up. There's two types of MHC. There's MHC1 and MHC2. Don't worry too much about the differences. They're used in different things. But here's where the genes are. They're found on chromosome 6. Oops, sorry. Found on chromosome 6. There's A, B, C, and D gene. A has all of these forms. B has these two columns of form. C has those different variations. And D has these different variations. So if you've got a set of these genes from mom and a set of these genes from dad, let's pretend that I'm A69 from my mom and AW34 from my dad, B14 from my mom, BW21 from my dad, CW7 and CW6 and DW5 and DPW4. What are the odds? What are the odds that I could find somebody with the exact same MHC? Unless I have an identical twin, and I don't. So, the MHC is the group of proteins found on your cell surface that identify you as you. So my MHC looks like that. And what happens is your T cells, now we're on the T cell side, your T cells go during fetal development. During fetal development and early in life, they go to the thymus. That's how they got their name. The thymus sits in the chest above the heart. They go to the thymus and they go to T cell school, where they read the book of you. So they read, my T cells went to T cell school when I was a fetus and early in life, and they read the book of Pam. There's the book of Pam. And they learned that the MHC complex, because this is all about MHC, they learned that my cell's MHC is this, and they should leave it alone. They fly around the body and they look around and they're like, oh, there's that Pam MHC, we're good, everything's fine. Now if I get, say, kidney transplant, liver transplant, does that organ gonna have the same MHC as mine? No. And this is where tissue typing used to be a big deal. So in order to get a transplant, you had to have an MHC match or have it close. You don't really do that anymore because the immunosuppressive drugs are so powerful. So there's no way. Let's say even if I was close. So it looks like this. Okay, that's my new kidney's MHC. So here's my T cell running around, looking around, saying, oh, that one's fine, but oh, that's bad, and I'm gonna kill it. And that's part of tissue rejection, right? Or organ rejection. Your immune cell sees that as foreign and destroys it. So that's why you need to take, if you have an organ transplant, a solid tissue organ transplant, you need to take immunosuppressive drugs forever. And those immunosuppressive drugs, basically, it's like putting dark glasses on the T cell so we can't really see. Yeah, that's Pam. Yeah, that's Pam too. That's Pam. We're good. Everything's good in here. Okay, right? You can't see. So they don't look at this and see it as foreign and they leave it alone. And people have to be on these powerful drugs for their life if they've got a solid transplant. So that's the T cell story. That's the MHC story. It's a complex protein group. We've got two kinds of MHC. Where am I here? Two kinds of MHC. MHC1 is found on all nucleated cells, and MH2 is found on your macrophages and antigen presenting cells. Don't worry too much about the differences. There's two different kinds. Just know that they're a complex group of proteins that identify you as you. Identify you as you. Okay, I'm putting there. I don't want to start this and then get halfway through it and not finish it. So, finish this on Tuesday. So get this under your belt now, at least through the first and second lines of defense. This is just an intro here, and we'll get through the rest of that on Tuesday. So get ready for test on next Thursday.